I'm Christopher Hill, and today I'll be presenting my summer research results for my project on nonsense-mediated mRNA decay, or NMD, in cardiac hypertrophy. So what is NMD? In the normal process of translation, a ribosome moves along an mRNA transcript, removing the exon junction complexes at the exon-exon junctions until it reaches the termination codon, at which point it disengages, and this results in the translation of a complete protein. However, in cases of mutation in which the termination codon is incorrectly placed 50 to 55 nucleotides before the last exon junction complex, the ribosome reaches the termination codon, which is now called a premature termination codon, or PTC, before it reaches the last EJC, exon junction complex. This results in nonsense-mediated mRNA decay in order to degrade this incorrectly transcribed mRNA transcript. So, the important proteins in NMD. UPF1, standing for up frame shift protein 1, is phosphorylized by UPF2 and 3. This phosphorylation results in the signaling of the mRNA degradation proteins used to actually perform nonsense-mediated mRNA decay. So these proteins, UPF1, UPF2, and UPF3, were our primary target proteins for studying mRNA NMD. So the role of NMD in the heart. Carrier and colleagues in Germany have shown NMD is involved in the pathogenesis of familial cardiac hypertrophy. However, no other studies have been done regarding NMD in physiology and the pathology of heart under other conditions. Hypothesis. Nonsense-mediated mRNA decay, or NMD, is regulated in disease-related cardiac hypertrophy and participates in disease pathogenesis. So, for our experimental approach, starting in vivo pathological hypertrophy, we used TAC surgeries, thoracic aortic constriction, studying the results after 2 days, 4 days, and 7 days post-surgery. The surgery being used to induce hypertrophy on the experiment hearts. We also studied the heart during developmental stages, the embryonic stages at 12.5, 13.5, and 15.5 days post-conception, and in postnatal models, 0 days, 2 days, 4 days, 7 days, and 2 weeks after birth. Switching to in vitro, neonatal rat and NRVMs, neonatal rat ventricular cardiomyocytes, we study the effects of UPF1 and UPF2 knockdown, as well as induced hypertrophy by phenylephrine and induced autophagy by starvation and rapamycin. We also studied the effects of mTOR activity under these conditions. So NMD gene expression in heart under pathological hypertrophy. These ANF and BNP are hypertrophy markers used to show the effectiveness of the TAC surgeries, all of which here were effective. And here we have the mRNA levels for UPF1, UPF2, and UPF3 proteins in these TAC hearts. Across the board here, we see a dip in NMD-related protein, in NMD-related mRNA transcripts at 2 days in each of these models. This dip is then restored in all models at 4 days, and it remains increased in UPF1. However, UPF2 and UPF3 again dip at 7 days. These results represent a very modest decrease in mRNA transcripts and require a significant amount more research for more accurate results. UPF1 protein is increased by TAC in heart. These results, on the other hand, are far more conclusive and powerful than the previous slides. We see at 2 days post-TAC surgery very little increase in mRNA NMD-related proteins. However, at 4 days, we see a drastic increase in NMD-related proteins here as opposed to here. And again, at 7 days, the hypertrophy models show much higher levels of NMD-related proteins in comparison to the control models. So this slide shows our evaluation of our UPF antibodies using siRNA knockdown in NRBMs. From this first band, we can see that our UPF1 siRNAs 2 and 3 were effective in knocking down the gene significantly. However, the first one was not. So we used these siRNAs for further experiments. We also see that our UPF1 primary antibody was effective in isolating the UPF1 protein bands. However, unfortunately, our UPF2 antibody purchased from Abcam was not effective and resulted in very large amounts of nonspecific binding. And we ordered replacements, but we were never able to find an effective UPF2 primary antibody. So we were not able to study the UPF2 for the rest of our experiments. So our experimental design for phenylephrine and NRBMs. On Wednesday, we plated the NRBMs. Friday, we introduced UPF1 knockdown by siRNA. On Sunday, we treated the cells with phenylephrine. The cells were harvested on Monday for a 24-hour exposure and on Tuesday for a 48-hour exposure. We then analyzed the mRNA with qPCR and the protein by immunoblot in NMD-related mRNA and proteins. So UPF1 gene expression by phenylephrine and NRBMs. In our plated cells, an exposure to phenylephrine in order to induce hypertrophy, we noticed a very modest, if even significant, increase in NMD-related proteins. This is consistent with later approved research. However, again, also consistent with later research, we noticed a much more significant increase in NMD-related proteins in response to phenylephrine treatment. So UPF1 knockdown showed a trend to enhance hypertrophic response to phenylephrine and NRBMs. Here we see that in the cases of UPF1 knockdown, response to hypertrophic induction by phenylephrine was higher in UPF1 knockdown, essentially in cells that were unable to perform NMD. The effect of phenylephrine, or the amount of hypertrophy induced, was, in most examples, significantly higher. This slide, however, shows the same experiments as the previous slide, but shows a much more modest increase in the hypertrophic response, and therefore I suggest that more research will be required in order to isolate the correct data. But as we stated, this is only a trend. So UPF1 knockdown, hypertrophic response by phenylephrine and NRBMs assessed by H3 leucine incorporation. So in this, we see again that although there was some experimental failure here, we noticed a very little increase in a hypertrophic response as a result of UPF1 knockdown. So experimental design for starvation or rapamycin in NRBMs. Again, we treated the NRBM, or we plated the NRBMs again on Wednesday, performed UPF1 knockdown on Friday, and starvation or rapamycin treatment on Monday for 4 to 6 hours. And we analyzed the proteins in NMD by immunoblot. So UPF1 knockdown increases autophagic flux. So as we can see here, in our control siRNA, the UPF1 knockdown that we can see is effective in here, here, and here all the way across resulted in possibly higher rates of autophagy flux, shown by LC3. But again, the increase is marginal at best and would require more experiments to further calculate the amount of effect. However, surprisingly, we tested for mTOR activity using PS6 and P4 EBP and found quite surprising results as a result of UPF1 knockdown. As we see here, UPF1 knockdown resulted in significantly higher levels of PS6, phospholipase 6 proteins, which are a downstream target of mTORs, and also some marginal increase in P4 EBP, which is also a downstream target of mTORs. Of course, the results would go on to starvation of vancomycin, or rapamycin treatments, which is standard. So, in summary, in vivo, we observed significant increases in NMD-related proteins as a result of TAC surgeries using hypertrophy on heart models, mouse models. In exposure to phenylephrine, we observed a significant increase in NMD-related protein concentrations. In response to UPF1 knockdown in NRVMs, we observed a possible increase in autophagic flux. And then most surprisingly, in response to UPF1 knockdown, our experiments showed an increase in mTOR activity. In conclusion, NMD participates in pressure overloading induced cardiac hypertrophy. Modulating of NMD, nonsense-mediated mRNA decay, may represent a novel approach to mitigate pathological hypertrophy and associated proteins disorders. For a working model, the understood pathway of pressure overloading resulting in mTOR activity, resulting in increased hypertrophy. However, our addition to this model suggests that pressure overloading may increase mRNA synthesis, which by nature of concentration increase would result in further rates of point mutations, resulting directly in higher rates of NMD. This may have a blocking effect on mTOR activity, which may then also have an effect on the amounts of hypertrophy. So for future directions, the findings of my experiments suggest possible connections between nonsense-mediated mRNA decay and mTOR activity, as well as autophagic flux. Further investigations into this link may yield very interesting and possibly productive results. However, further analysis of NMD rates could be provided using more indicator proteins such as UPF2 and UPF3, as well as studying the rates of phosphorylation on UPF1. Possible means to enhance NMD and assess pathological hypertrophy in vitro, and considering knockout of key NMD molecules in vivo and examine pathological hypertrophy and functional consequences. So for my acknowledgements, I'd very much like to thank the whole lab, especially Zhao. Thank you so much for helping me through this project, being patient with me and all my errors, but also everybody else for being very supportive and understanding and guiding me through this.